Okay, let's continue to Meiosis II. Meiosis II, we start with this. We start with prophase 2. So nucleoli and nuclear membrane disappear. Okay, so same. Okay, you see the events that happen in prophase 1, in prophase 2, and even in prophase in mitosis, same. So each chromosome is made up of sister chromatids that are joined at the centromere. So now it is referring to the events that happens in these two daughter cells, two haploid daughter cells. So still spindle fibers starts to form in both daughter cells. Okay, moving on after prophase is metaphase. So again the term equatorial plane is here. So you have to draw dotted lines. So notice that these chromosomes here, one is shorter, the other one is longer. They are not homolog to each other. Okay, so when you study this part here, meiosis, actually you have to use your pencil, a paper, you have to draw, you have to sketch, then only you can remember, right? So hopefully you'll do this at the same time, okay, or afterwards. Okay, so now they are not homolog to each other because here in anaphase 1, the homolog already separated. Okay, so in metaphase 2, chromosomes are arranged. So chromosomes, the word chromosomes are highlighted. So maybe they want to emphasize or to remind the students that it is not the homologous chromosomes unlike in metaphase 1. So arranged at random. Okay, this is important because of variation. If they arrange randomly, then different variations will happen. Imagine this is just telling you the story if the parent cells are having 4 chromosomes. But imagine human beings, how many chromosomes do we have in total? Yes, 46. So the variation is very large. Okay, so back to metaphase 2, each chromatid is tied to the spindle fibers at the centromere. Okay, so metaphase 2 ends when the centromere separates. Okay, so here separates. So the sister to one another, the sister chromatids is now separated. Moving on, next is anaphase 2. So the sister chromatids centromere starts to separate. Unlike here, anaphase 2, the centromere is not separated and the sister chromatids are still tied to the same centromere. Okay, so in anaphase 2, sister chromatid pair separates and they are moving to the opposite poles by the contraction of the spindle fiber. Okay. Next is telophase 2. So from here, you can see, remember, when you draw, make sure that the centromere is leading to the pole, okay, not the legs, okay, because this part is being pulled. So if you draw reverse like this, okay, it's going to be penalized. So in telophase 2, chromosomes appear at the pole of the cell. So spindle fiber disappears, the rest are the same, nuclear membrane, nucleoli reconstructed. But look here, the number of chromosomes for each daughter cell. So how many daughter cells now? You can see from the picture, 1, 2, 3, and 4 daughter cells, right? And each daughter cell is having half the number of parent chromosomes. So let's confirm by counting the centromere. 1, 2. Okay, so each cell having 2 chromosomes. You look at the parents. 1, 2, 3, 4. So confirm that it is haploid. 4 daughter cells and they are haploid. So each haploid cell contains half the number of the parent cells. And the genetic content is also different, okay, from the diploid parent cell. Okay, and the haploid cells eventually they will develop into gametes, and this is actually the process of gametogenesis. All right.